Thank you for joining us for today's webinar. My name is Cassie, Marketing Supervisor at GenScript, and I'll be the moderator for the webinar today. So a little bit background about GenScript for those who are new to us. GenScript is the leading life sciences research and application service and product provider in various fields, from basic life sciences research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. GenScript ProBio is the biologics CDMO segment of GenScript, proactively providing end-to-end service from drug discovery to commercialization with proactive strategies, professional solutions, and efficient processes to accelerate drug development for customers. The topic for today's webinar is high-quality plasmid DNA manufacturing strategies for gene and cell therapeutics. So let me quickly introduce our speaker for today, Dr. Ming Ni. Dr. Ming Ni is the manager of CMC Project Management Department in GenScript ProBio. He received his doctoral degree from the University of Washington, and he joined GenScript in January 2020. He has been responsible for technical support of Plasmid DNA platform, and he led the Plasmid DNA project to improve the yield and optimize purification steps. Before he joined GenScript, he was a full professor and department head of biomedical engineering department at Yankee Tech University, with over 50 peer-reviewed articles, 1,600 citations, and 4 US patents. He is one of the most prolific scientists at GenScript. This webinar will take around 45 minutes, and we'll have about 15 minutes for Q&A session after the presentation. You can type in your questions in the chat box, and we'll answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we'll email back the answers to you after the webinar. So now, without further ado, we'll let Dr. Ming Ni to start his presentation right now. Dr. Ming Ni, over to you right now.
Thank you, Cassie. Good afternoon, everyone. And today is my pleasure to talk about my presentation and to talk about the high-quality plasmid DNA and manufacturing strategy for gene and cell therapy. Okay, for today's talk, I will divide it into 4 sessions. The first one, I will talk about the plasmid DNA from market perspective. And you will see that the market for plasmid DNA is really increasing from year to year. And the second one, I will talk about why plasmid DNA matter. Yeah, can you go back 1 slide? It's going so fast. Yeah, thank you. Yeah, the second part, I will talk about why the plasmid DNA matters. We will use it for different applications, especially to make lentiviruses for CAR T cell therapy, to make AAV particles, to make mRNA vaccines, and even plasmid DNA itself. It can be used as a DNA drug or DNA vaccines. The third part, I was talking about the manufacturing strategies, and I will talk about how we produce those plasmid DNAs at GenScript ProBio. By the way, our location is at Zhenjiang, China. Last but not least, I'll talk about the GenScript ProBio one-stop solution. Yeah, thank you. Next slide, please. Next slide. Yes. From these slides, you will see we summarize all the global marketed advanced gene and cell therapeutics. And you will see that from 2005 to 2016, over 10 years, only 3 drugs have been approved by FDA or other regulatory agency. And after that, we've seen 4 or 5 years, starting from 2017, you will see more than 9 drugs have been already approved. So this really means that gene and cell therapies is really booming for the coming years. Next slide, please. Okay, in this slide, you will see the global market for plasmid DNA. In the right-hand side, you will see the pie chart. For year 2019, the global market size is about $1 billion for both viral vectors and the plasmid DNA. And for plasmid DNA is about 13%, 20%. For the outer pie, you will see that is the prediction for the year 2030. By that time, the global market for both viral vectors and plasmid DNA will be 4.8 billion US dollars. Although the plasmid DNA is only contribute 13%, but the absolute value, you will see that from the right-hand side, the chart, starting from 2019 is about $200 million. And by the year 2030, this number will be increased to $780, $80 million. This is clearly show that the trend for plasmid manufacturing, the market is really booming. Okay, next slide. So in the second session, I'll talk about how this plasmid function. Next slide. Yeah, for the plasmid DNA, it can be used for gene and cell therapy. And for example, the CAR T cell therapy and gene therapy. I will talk about more in the details in the next few slides. Next slide, please. Yeah, these slides, I will not go to the details, but you can see that use plasmid DNA, you can use it for packaging lentivirus and AAV, adeno-associated viruses. And for the lentivirus, we call it, in short, LVV. We use third generation 4-plasmid system, and the 4-plasmid can co-transfect HEC293 T cells and to produce lentiviruses. For the AAV, typically people use 3-plasmid co-transfected HEC293 cells, and you can get the AAV. In this case, you can get AAV2. Next slide, please. Yeah, the messenger RNA vaccine is getting very popular in this year. And you will see that messenger RNA really need plasmid DNA as a template. And then you do the IVT reaction, in vitro transcription reaction, and you can get the messenger RNA. The IVT messenger RNA has a structure showing here. It composed of 5 different parts. You have 5' cap, and you also have the 3' end. You have poly-A tails. And in the center part is open reading frames. This part is very important because this part typically is coded for proteins of interest, especially like for COVID-19, and you can code it for spike protein, glycoproteins. And of course, 5' UTR and the 3' UTR region, they are also very important. In this schematic diagram, it's coded for light green and dark blues color. Those are very important because designing the messenger RNA molecule is very important for the success of messenger RNAs. As many of you know that CureVac, one of the giants in the messenger RNA industry, recently their phase 3 clinical trial didn't show successful results. That, once again, it shows that it really matters. And the major difference between CureVac and other messenger RNA company like Moderna or BioNTech, because CureVac, they didn't use chemically modified nucleotides. That's one of the major difference. Okay, the right-hand side shows that the schematic diagram, how the messenger RNA works. So you can see that 2 types of messenger RNA, either the non-replicating messenger RNA, in short NRN, or the self-replicating messenger RNA, in short SAN. They can be packed into the drug delivery vehicles. Typically for the messenger RNA, the commercial approved system using LNP, the lipid nanoparticle, to deliver messenger RNA into the cells. Once they cross the cell membrane, you can see that the messenger RNA has been released from the delivery vehicle and being translated into the protein of interest. And then later the protein either being secreted by cells, and they will be recognized by B cells or T cells, and that will initiate the immune response. Of course, this is the short version. If you are interested, you can look into many review papers, and you can understand a little bit better about how this messenger RNA vaccine works. Next slide, please. Okay, then about the DNA vaccine. DNA vaccine, from this diagram, you can see that it's different than the messenger RNA because it needs to go through two barriers instead of one. It has to go cross the cell membranes and the nuclear membranes. Once you enter the cell nucleus, it can, according to the central dogma, everybody knows about it, and DNA can be changed to the messenger RNA, and later messenger RNA can be translated into peptides or proteins. And these peptides, either those transmembrane peptides or secreted peptides coded for antigen, of course, then be recognized by B cells to initiate the immune response, or they can be presented at the cell surface as part of the MHC, either type 1, class 1 or class 2, and this will be recognized by the T cells and also will initiate the immune response. This is, of course, a very brief, simple version. The DNA vaccine works is quite, how to say, difficult compared to the messenger RNA because it's not easy to highly efficiently deliver the DNA and the plasmid DNA into the cell nucleus. So this is one of the challenge you do that. Okay, next slide, please. So in the third part, I will talk about the manufacturing strategies of plasmid DNA. Basically, for the Zhenjiang site, we use this strategy to produce plasmid DNA. This diagram show is the flow chart, show that how we can produce the plasmid DNA. You can see starting from fermentation, we go through cell harvesting, lysis, purification, concentration, go through three steps of chromatographic purification, then we go concentration, and later fill and finish, you can get your final products. So this is quite a robust system, and we have done many batches and support many IND application to be successfully get the IND approval. Next slide, please. Okay, from the last diagram, actually, you can see that typically we divided the whole process into upstream and downstream. The upstream optimization you can do starting from carefully select the culture media. Based on different host strain, you can use different culture media, and different company have their secret recipe to get the really high yield of plasmid. Starting from screening the culture media, you will find the optimal media for E. coli strain to grow, and later you can confirm this culture condition using shake flask. And after you get the roughly the idea how you are going to culture them, you can move everything to the bioreactor, or you call it a fermenter. Then you can optimize many parameters, including the feed, feeding strategy, pH, other parameters like inoculation, dissolved oxygen, temperature, and so on and so forth. And last but not least, you can optimize the harvest time. Okay, next slide. After the upstream optimization, here I show you just a case study. The pH is one of the important factors. As you simply change the pH from one condition to the other, you can increase the plasmid yield. Another important factor is carefully choose the whole strain. As many CDM company, they don't have our GenScript system. We have more than 7 commercial whole strain, E. coli strain you can choose, and this can really boost the yield of plasmid. On the right-hand side, you will see the results of upstream fermentation. And in the first diagram, you see that in red, you have OD 600, which directly proportional to the number of E. coli. And you have the green colored curve that's representing the yield of the plasmid. And very importantly, you see the purple line. The purple line really coded for the unit yield. This is milligram per liter per OD. So this is very important information for you when you want to optimize your upstream process. Another important results we got is when we use 2 different scales, like 5 liter or 10 liter, we can get pretty much similar production. That means the scalability of our process is really good. Of course, right now at the Zhenjiang site, we can easily to manufacturing plasmid DNA up to 100 liter without any problems. And the take-home message down there, you'll see that now we are proud to claim we can produce 1 gram per liter plasmid, which is much better compared to many companies in the market. You can find out this is a really high yield. Okay, thank you. Next slide. Yeah, for the downstream, it's quite important to, once you have the plasmid DNA product, you want to remove all the contaminations. But to remove all this contamination, you need to understand very well about the plasmid DNA. See here, I list the structural and the chemical characterization of plasmid DNA. This is the foundation of the basic information, you know, how you separate your plasmid DNA, your product, from other contaminants. For example, on the right-hand side, you see the table summarize some impurities commonly you will encounter in the process optimization. The plasmid DNA has different isoforms. For example, you have linear form, you have open circle form, and the supercoiled plasmid DNA. The supercoiled plasmid DNA has already been proved. This is the most functional one you use as a DNA drug or vaccine. So this is the product you really want. But as you can see, all these isoforms, they share the same size, and the only difference, they have the charge different. Then you can use affinity chromatography to separate that, or you can use HIC, is hydrophobic interaction chromatography. Another example is protein and DNA. Because the protein and DNA, they have different size. So based on the size difference, you can use SEC, size exclusive to separate the proteins from your product. And very commonly, you will run into the impurity is RNA. The RNA and plasmid DNA, you can separate that either use the charge, the difference, the RNA and the plasmid DNA have a different charge, or they have a different hydrophobicity, or simply by the size. And from the based on the different parameters you choose, you can choose accordingly the chromatography columns to separate RNA from your product. Okay, next slide. Yeah, this slide I want to show you that for the downstream process, we get the pretty good results. For 10-liter batch, we can get the plasmid DNA yield above 800 milligram. Many cases we get over 1 gram without any problem. For the recovery rate, because of the project are quite different, but we are able to get the recovery rate above 20%. Usually the recovery rate is between 20 to 60%. And the benefits using the GenScript ProBio system, the downstream process, is we get the very high purity of the plasmid DNA product. As you can see, the high purity, one of the indication is IEC percentage. IEC percentage is corresponding to the supercoin content. If this content is close to 100%, that means your product is very pure. HCD is whole cell DNA, HCP representing whole cell proteins. Those numbers you can see that is very, very low. It's below 0.01% in many cases. So that means our plasmid DNA has very high purity, so-called, we call it high quality plasmid DNA. Okay, next slide, please. So this table shows the release criteria of our plasmid DNA. We have excellent QC and QA team to guarantee our customer to get the high quality plasmid DNA. We have done many assays to show that the purity as indication for supercoin content. We use HPLC, for example. We can get the purity above 90% without any problem. And for the impurity, like I mentioned before, the host protein, the residue E. coli DNA, those are very, very low. And the RNA, as I mentioned in the previous slides, those commonly encountered impurity. And from our release, COA, we call it, you can see that typically we cannot detect any RNA residue in the product. And very importantly, when you use our plasmid DNA for clinical study, the endotoxin level is very important parameters, and we can get less than 10 EU per milligram. So this is way better than the FDA the guideline. This guarantee our plasmid DNA has high purity, and this is the advantage over many other companies. Okay, and of course, many of our customers want to get the sequence of our plasmid DNA product, and we typically use Sanger to do the DNA sequencing. And of course, based on your request, we can use other up to now the technology to do the sequencing for you, because we get more requests for the DNA template for mRNA projects. Okay. Later we can discuss if you have any special request for the QC release standard. Okay, next slide, please. Yeah, these slides, we summarize some results we got previously up to March 2021. And we have done, since then, we have done more batches for messenger RNA. We typically provide the template DNA for messenger RNA vaccines, and we did the process optimization. As for one project, 50-liter scale project, we can optimize the DNA yield from without optimization, and after optimization, we can get 4 to 5 times increase for the yield. This is quite important. And also, very importantly, our process is quite stable, and we don't have a very high batch-to-batch variation. As you can see, we done, for example, project B and C, we don't see the yield is different from the time to time when we make the manufacturing. And poly A tail, typically people will include that as I illustrated in the previous slides. This is very important to make your messenger RNA stable, and also later for the translation part work. And we have done poly A tail from 80 nucleotides to 150 nucleotides. We can do it easily. Yeah, go back to the slides. Yes, okay. So we can probably see we can do this DNA template for messenger RNA with success rate very high. Okay, next slide, please. Yeah. Here, I want to switch gear a little bit, talking about plasmid for lentivirus packaging. For the lentivirus packaging, we have ready-to-use lenti-helper plasmid, as listed here. Those are already being registered by the CBER FDA, and this is good for your IND application, because all this plasmid has already been registered and listed in the FDA dossiers. And if you submitted your IND application, the FDA will not looking into the details for those helper plasmid, because these are already been proved they are safe to use in other projects. So it's really good for our customer. You can directly use those off-shelf, ready-to-use helper plasmid. And on the right-hand side, it shows those helper plasmid really help for the packaging efficiency for both suspension culture and adherent culture. We can get very high titers of lentiviruses. Yeah, next slide, please. Yeah. Now I want to talk about the GenScript ProBio. Yes, thank you. GenScript group, actually we have 4 different business units. The first one is life science ARO. The second is Best Design, is mainly focused on the industrial synthetic biology products such as enzymes. And the third business unit is Legend Biotech. It focuses on the cell therapy, especially CAR-T cell therapies. It's already went to the US stock market, achieved success, clinical results showing very promising to get the BLA possibly this year. Okay. Last but not least is our business unit. It's called GenScript ProBio. Next slide, please. GenScript ProBio, our vision is your most reliable and sustainable biologics partner. So our mission is dedicated to proactively providing end-to-end service from discovery to commercialization with professional solutions and efficient processes to accelerate drug development for customers. Yeah, next slide, please. Here, I want to show you the global partnership and a solid track record of GenScript ProBio. Over the last 2-3 years, we have done 60 plasmid and virus CMC projects and clinical GMP projects. 9 of them already get the IND approval, either from PMDA or FDA or NMPA. Among these 9 IND approval projects, 4 are mRNA projects. And we also done 20 over plasmid virus CMC projects. We have done over 30 clinical plasmid manufacturing batches. And also we have done plasmid and CMC manufacturing for over 10 messenger RNA customers. So here we show the key account. We actually very proud to say that we provide plasmid and lentiviruses for over 30 big pharmas and leading biotech companies. And down to this slides, we show the three very successful story. We have done collaborating with X Life Science and also STEMI RNA and Aborigine. We help them to get IND approval. Okay, next slide. So this slides our RA department summarize the plasmid based on the application. For example, for the cell therapy, if you use plasmid for CAR T cell therapy, those plasmid are considered, the transfer plasmid are considered as intermediate. And then you should consider using GMP principles. And for messenger RNA products, and the plasmid DNA is used as a template for messenger RNA. Then the plasmid DNA is considered as raw material. Once again, you use it under the GMP principles. Down there, we have 3 different grades of plasmid. I'll talk about more in the next slides. Next slide, please. So for GenScript ProBio plasmid one-stop solution, we have 3 different grades, so-called Pro grade, GM Pro grade, and CGMP grade. The Pro grade plasmid is superior solution for preclinical studies, research and discovery stage, for R&D stage. This is sufficient for your research. And typically people come to us to do CMC projects that's aiming for the IND filing. And for from IND filing up to the early stage clinical trials like phase 1 and phase 2, you can use GM Pro grade plasmid. For the DNA drug or vaccine, you can use so-called clinical GMP grade. This is equivalent to GM Pro grade. For phase 3 BLA application and later stage commercialization, you can use CGMP grade plasmid. Down there is summarized for different grade, suitable for different stage of studies. You can use this as a reference. Next slide, please. Yeah, to summarize the today's presentation, I want to say using GenScript ProBio plasmid, it balances the cost and compliance. GenScript ProBio offers one-stop solution to support various applications with a strong track record. We have done various applications. As you can see, we achieved 9 IND approvals, including CAR T, TCR-T therapy, CRISPR messenger vaccines, DNA vaccines, or we can also produce LC DNAs. So far, we get 9 IND approvals by US FDA, Japan PMDA, and the China NMPA. Yeah. So for the technical advantage, the plasmid for lentivirus or AAV, we have a ready-to-use lenti-helper plasmid, as you remember. And we also achieve very high density fermentation. For the upstream, we can get plasmid yield over 1 gram per liter. And for plasmid, for messenger RNA, we have done over 10 batches of plasmid with length of poly-A longer than 100 BP. The longest one we can get, the poly-A tail is around 130. For the DNA vaccines, we can also achieve very high concentration, up to 20 milligram per milliliter. And also we have fast turnaround time. Within 6 to 8 weeks, you can get the product. And for the cell modification, so far we have done 3 projects for LC-DNA. This is super for engineering your cells. Okay, next slide. Yeah, thank you very much for your attention. And if you have any question, please let me know. I'll be happy to answer them. Yeah. Thank you.
Thank you, Dr. Ming Li, for the great presentation. We are open for questions right now. So attendees, you can actually type in the questions in the box on your screen. If there's no questions from the floor, we'll close the session today. But feel free to email to us if you have any questions after the session. We'll get back to you via email. So thank you once again, Dr. Ming Li, for the presentation, and to all of you who joined us today. I hope you find this session beneficial to your work. Thank you and have a nice day. We have one question from the floor. Just hold on. We just got one question from Chen Yunzu. The question is, why is your IVT RNA synthesis service stopped recently? Hi,
Can
Dr.
you repeat the question?
Yeah, sure.
Can you repeat the question?
The question is, why is your IVT RNA synthesis service stopped recently?
Okay. Actually, we provide the plasmid DNA. We provide DNA template. We don't provide messenger RNA directly.
Okay. I hope that answers your question, Yunzu. So if there's no other questions from the floor, we'll close the session right now, but feel free to email to us if you have any questions after that. So thank you once again and have a nice day, everyone. Bye-bye.